Hello everyone, welcome to the new session of seed storage. So in this session we are going to study about what is seed storage. So this is the first topic, and then there will be an introduction to the seed storage. Stages of seed storage. So there are different stages that we are going to study about. Purpose of seed storage, why we store the seeds, right? Storage at the ambient temperature and humidity. So these are the different methods of seed storage. So the first one is the storage at ambient temperature and humidity. Second, dry storage with control of moisture content but not the temperature. So in this topic we are going to study about the storage of seed, that is dry storage, and the control will be just for moisture. There is no control for the temperature. Another method is the dry storage for the long-term gene conservation. So this is the method that we follow to store seed for the long term, right? And that is for the gene conservation. Next method is the moist cold storage with control of temperature. So in this topic, we are going to study about the control of temperature on seed storage, right? And the last one is that is the cryopreservation. So these are the different topics that we are going to study about in this particular session. So let us start. Preservation of seed with initial quality until it is needed for planting. So the seeds that we get initially from the crop after the crop harvesting, that is called the initial quality of the seeds, and we want to store that seed in that form only, right? So the preservation of seed with the initial quality until it's needed for planting. So for that we need seed storage. The ability of the seeds to tolerate moisture loss allow the seeds to maintain the viability in dry state. So this is the capacity of a seed, that when there is a no moisture available to the seed, okay, so they can tolerate the moisture loss and allows the seed to maintain being viable in that dry state also. So storage starts in the mother plant itself when it attains the physiological maturity. So when the seeds are formed and when the seeds are at the mature form in the when they are attached to the mother plant only, then only they are meant for the storage. Okay, so the seeds start storing its embryo in that form only at that particular time. The seeds are considered to be in the storage form the moment they reach the physiological maturity until they germinate to until they are thrown away because they are dead or otherwise worthless. So only when we are going to germinate, that seed goes to germinate, means it comes out of its the dormant state and it give out its plumule and radicle and set up in a plantlet. But sometimes what happen, the seeds are lost. The seeds are lost because the seeds are not viable, so we throw those dead seeds, okay, because they are worthless. The entire storage period can be conveniently divided into the following stages. So we are having different stages, and the period is decided of its storage accordingly. So, stages of seed storage. Storage on plants, physiological maturity until harvest. So, the seeds store the baby plant in that, and that is at the physiological maturity. Harvest, until the process the store in a warehouse. So, when it get processed, it is harvested seeds are stored in the warehouses. In storage, so properly we store those seeds in warehouses. In transit, railway wagons, trucks, carts, railway sheds, etc. So the large amount of seeds are transported from one place to another, and for that also for some time they are stored at the railway wagons, in the trucks, in the carts, railway sheds, etc. In retail store also the seeds are stored for longer time, right? And on the user's farm. In fact, in our home also we collect the seeds and store it properly, right? And we keep some of the things like that could not harm those seeds. There are many chemicals, there are some antibacterial or anti insects material, like some of the plants, like neem plants. Okay, the leaves are kept with the seeds so that they do not get any type of harm or any type of infection with the fungi, bacteria, or even insects. Now, introduction. The ability of seed to tolerate moisture loss allows the seeds to maintain the viability in dry state. So the seeds are viable in dry state and they can tolerate the moisture loss. So this is the seed that is able to withstand this adverse condition. The storage start in the mother plant itself when it attains the physiological maturity. So when the seeds are set up, when the seeds are formed, when they reaches to the physiological maturity, they start storing the embryo that is present inside the seed. Okay. So after harvesting, the seeds are either stored in the warehouses or in the transit or in the retail shops. So there are three places that they can be stored. In the warehouses, they are stored there for the longer time, and in the transit also. And the third place, that is at the retail shop. So after harvesting, the seeds are stored at these places. During the old days, the farmers were used to farm safe seeds in little quantity, but introduction of high yielding varieties and hybrids and modernization of the agriculture necessitated the development of storage techniques to preserve the seeds. So earlier what happened? The farmers used to store the seeds. Okay, so there are some material, that is boxes, wooden boxes, or even the cart that is made up of mud. They store the seeds in that, okay, for their use. But the larger quantity that were stored in the warehouses. But nowadays we are having such a new storage techniques to preserve the seeds for the longer duration of time. The practice of storing the seeds start from the ancient days itself. So this is not a new thing that we store seeds for our whatever the time we need. We can use the seeds in our near future. So we are doing from the ancient days itself and following the simple and the cheap techniques. For example, placing the seeds in salt. When you place the seed in salt, it do not get infected with the fungi, bacteria, or any other insects. Red earth treatment to red gram. But the same practice are not hold good for the present day agriculture. Okay, so this is the one of the method to store the seed, but for all the seeds this is not applicable, because large quantity to be stored, we have to store a large quantity of seeds, so we cannot waste so much salt for that, right? Exchange of varieties and species and exchange of genes. So there are so many things that we need to consider for the seed storage. The type of material to be stored decide the techniques to be followed for the safe storage. So we are having different things to store. We are having the tubers, we are having the fruits, we are having the fruits and vegetables, we are having dry seeds. So we are having different things to store, and for every material, for every type of fruits, vegetable and seeds, we are having the different techniques to store. Nowadays, storage technique changed from the ordinary go down storage to cryogenic tank storage and even gene storage. So earlier we are just having the go down storage. We stores the our seeds in go down. But nowadays we are having cryogenic tank storage. This is more applicable and more beneficial. And even we are having gene storage also, because we are having so many new varieties, right? So for that we require gene storage. To maintain initial seed quality, that is germination, physical purity, vigor, etc., all along the storage period by providing suitable or even better condition. So we need to maintain the seed quality. Means that whatever the method we are following for the seed storage is should not harm the quality of the seed. And what are the quality of seed? The germination of seeds, physical purity of seeds, vigor of the seeds, okay, and all along the storage period by providing suitable or even better condition. So whatever the method we are following, it should not affect the seed quality. Since the main objective of the seed storage is the maintenance of an acceptable capacity for the germination and emergence, it can only be accomplished by reducing the rate of deterioration to the degree required to maintain an acceptable level of quality for the desired period. So what is the main objective of our seed storage? The main objective of our seed storage is to maintain an acceptable capacity, okay, maintain an acceptable capacity for the germination of seeds and emergence of the new baby plant. Okay, it should be reduce the rate of deterioration. Our seed storage method should be like that it should not deteriorate the rate of deteriorate the rate of seeds germination or the storage degree required to maintain an acceptable level of quality for the desired period. So whatever the desired period we want to store a seed, that should be like it should maintain the acceptable level of the quality. Means for that duration of time, the quality of the seed should be maintained. So these are some of the main objective of our seed storage. Seed storage is the maintenance of high seed germination and the vigor from the harvest until planting. So it should maintain the seed germination capacity. It should maintain the vigor from the harvest. Okay, so the seeds are in a good condition when they are harvested, suppose, but it should not lose its vigor, it should not lose its seed germination capacity after the seed storage. So that method we should adapt for the seed storage. So that is very important. It is important to get adequate plant stand in addition to healthy and vigorous plant. Every seed operation has or should have a purpose. The purpose of seed storage is to maintain the seed in a good physical and physiological condition from the time they are harvested until the time they are planted. So from the time of harvesting and for the time of planting, in the mid of this, we store the seed. And this method, whatever the method we are applying for the seed storage, it should maintain the good physical and physiological condition of the seeds, right? It should not damage the seed's vigor. It should not affect the seed germination capacity. Otherwise, the seed will not germinate and that seed will be of no use. Okay, so this is the physical and maintenance of the physical and physiological condition of the seed, and that is very important in our seed storage method. The seeds have to be stored, of course, because there is usually a period of time between harvest and planting, right? And for all over a long time, we require that seeds for our food, right? So we have to store these seeds, and during this period the seeds have to be kept somewhere, while the time interval between the harvest and planting is the basic reason for storing seeds. And there are other considerations, especially the case of extended storage of seed. So there is harvesting of seeds, there is a harvesting of seed, and there is a planting, new plant from these seeds. Okay, so every seed has its own requirement for the condition required for the planting, right? There may be moisture, there may be availability of soil, there may be the availability of weather condition. So we are having kharif crop, we are having rabi crop, we are having zaid crop according to the season. So in that particular season, that seed will grow. So when they are harvesting, they are stored for a time. They are stored for a time period. And after, when the season is again back, right, the next season, we take those harvested seed and go for its germination and later planting that seed in the cropland. So for a time duration we store that seeds, right? So that is why the storage of seed is very, very important. Seed suppliers are not always able to market all the seeds they produce during the following planting season. In many cases, the unsold seeds are carried over in the storage for marketing during the second planting season after the harvest. So, seed suppliers, what they do, they store the seeds, okay? And that stored seeds can be used for all over the year. And they also store some of the seeds for the next season for planting. Okay, so this is the uh, use of the seed storage. The problem arises in the connection with the carryover storage of seed, because some kind varieties and lots of seeds do not carry over very well. So if we are not keeping the seeds in a proper way, so they cannot be stored for all over the year, and that can be a loss, a great loss to the farmer, a great loss to the storage person. There are different we can say factories that stored the seeds, okay? And if the seeds are not stored in a proper required condition, then there will be a loss of those seeds, okay? Loss of those seeds. So it is very, very important to store the different varieties, different kinds of seeds according to their storage, right? So seeds are also deliberately stored for the extended period, so as to eliminate the need to produce seeds very soon. Okay, so these are some of the purpose of seed storage, why we are storing the seeds. Foundation Seeds Unit and other have found this to be an economical, efficient producer procedure for the seeds of varieties for which there is limited demand. Some kind of seeds are stored for the extended periods to improve the percentage and rapidity of germination by providing enough time for a natural release from dormancy. Regardless of the specific reason for storage of seeds, the purpose remains the same, that is, maintenance of a satisfactory capacity for the germination and emergence of the baby plant. So these are the two important aspects, right? Regardless of the specific reason for the storage of seed, why we are storing the seeds, the purpose remain the same, to maintenance of a satisfactory capacity of the seed germination. The seed should germinate. After germination, the new baby plant should emerge out of the seeds. This is the main motto of seed storage. Regardless of the method we are following for the seed storage, the facilities and procedure used in seed storage therefore have to be directed toward the accomplishment of this purpose. In the broadest sense, the storage period for the seed begins with attainment of the physiological maturity and ends with the resumption of activity growth of the embryogenic axis, that is germination. Seeds are considered to be the physiologically and morphologically mature when they reach maximum dry weight. So, when the seeds are set up, they attain a physiological maturity, they attain a morphological maturity, and when they reaches to the maximum dry weight. So that shows that this is the uh, time that we must store the seeds. Okay. At this stage, dry down or dehydration of the seed is well underway. So this shows that all the water, all the moisture that is available in the seeds, now that has been dried up. Okay, so at this stage, the dry down or the dehydration of seed is well underway, and the dry down continues after the physiological maturity until moisture content of the seeds and the fruit decreases to a level which permit effective and efficient harvest and threshing. So this is very, very important that at the time of seed storage, there should not be any moisture present inside the seeds. If it is present, then it can harm to the seeds. It can destroy the seeds. So all the seeds that are going for the storage should be dried up. Okay, the moisture content of the seed should be decreased. The fruit, if we are storing, then also it should be decreased. This storage can be termed as a harvest maturity. So there usually is an interval of time between the physiological maturity and the harvestable maturity, and this interval represent the first segment of the storage period. So we are having an interval time, that is reaching of the seeds at a physiological maturity and harvestable maturity. So in between these two, there is a first segment of the storage period, means this is the time that we can store the seeds. Any delay in harvesting the seeds after they reaches the harvest maturity prolongs the first segment of the storage period. So if we delay the harvesting of the seeds, then it can prolong the first segment of the storage period and offer determinant of the seeds quality, and that can affect the seed quality. So the harvesting must be done at a proper time. Storage at the ambient temperature and humidity. So seeds can be stored in piles, single layers, sacks, or open containers under the shelter against rain, well ventilated and protected from rodents. Store at least for the several months. So seed can be stored in piles, in single layers, or even sometime in the sacks or open containers. But it should not be exposed to rain, and it should be well ventilated, and there should be protection against the rodents. So that can be stored for months. So this is called the storage at ambient temperature and humidity. Dry storage, now that earlier what in the previous slide we have studied, that was not a dry storage. Now dry storage with control of moisture content but not the temperature. So the orthodox seeds will retain viability longer when dried to the low moisture content, that is 48%. So the content of the moisture is reduced to a great level and then stored in a sealed container. Now this time the container are not open, or in a room in which the humidity is controlled, and you can store it in a room also in which you can maintain the humidity. So control then when stored in the equilibrium with the ambient air humidity. So cool condition is especially favorable. This is recommended for many orthodox species which have periodicity of seeding but which are planted annually in large-scale afforestation projects. A combination of 4 to 8% moisture content and 0 to 5% temperature will maintain viability for 5 years or more. So, for a longer duration of time you want to store, then you can have this much availability of moisture content. You should maintain the moisture content to 4 to 8% and the temperature, that is 0 to 5%. So this at this condition, this seeds can be viable for 5 years or even more. Long-term conservation of gene resource of the orthodox agriculture seed is at minus 18 degrees centigrade temperature and 5 to 11% moisture content. Moist storage without control of moisture content of the temperature. So suitable for the storage of recalcitrant seeds for a few months over winter. So this method is applicable for the recalcitrant seed, not for the orthodox seeds. So seeds may be stored in heaps on the ground, in shallow pits, in well-drained soil, or in the layers in well-ventilated sheds, often covered or mixed with the leaves, moist sand, peat, or other porous material. So some of the material like porous material we can use for its storage. We can mix it and we can store for this. And we can also keep it in a heap on the ground. The aim is to maintain a moist and cool condition with good aeration to avoid overheating, which may result from the relatively high rates of respiration associated with the moist storage. So this may be accomplished by the regular turning of the heap. So you should uncover the heaps. You should regular turn over the heaps, okay, so that the moist can be available, cool condition can be available for the seeds. Moist cold storage with control of temperature. So this method implies controlled low moisture just above the freezing, or less commonly just below freezing. So we are maintaining the temperature just below the freezing and just above the freezing. Okay, so the moisture can be controlled with approximate limit by adding moist media, that is sand, peat, or the mixture of both to the seeds. So in this way we can store the seeds in proportion of 1 part media and 1 part seed by volume, and re-moistening periodically or more accurately by controlling the relative humidity of the store. So this method is much applicable to temperate recalcitrant genera. So this is very, very important, which this method is used for which type of seed storage. So temperate recalcitrant genera, we can go for the moist cold storage with control of temperature. Okay, next is the cryopreservation. This is also called the cryogenic storage. Seeds are placed in liquid nitrogen at minus 196 degrees centigrade, very low temperature. Seeds are actually placed into the gaseous phase of the liquid nitrogen, that is 150 degrees centigrade, for easy handling and safety. Metabolic reactions come to the virtual standstill, and at the temperature of liquid nitrogen, the cells will remain in an unaltered state until the tissue are removed from the liquid nitrogen and defrosted. So basically, they are frosted at that particular time. And when we are going to use the seed, then we have to defrost it. Therefore, the little detrimental physiological activities take place at these temperatures. Very less physiological activities are taking place, and which prolong the storage life of the seeds. It is not practical for the commercial seed storage, but it is useful for the storage the valuable germ plasm. This method is only used for keeping a valuable germ plasm, right? This is not used for the commercial seed storage. For the germ plasma storage, we can go for the cryopreservation. Okay, so this is all about this session. With this session, we have completed the different types of storage for the seeds. Thank you very much.